by protein modeling and binding ability. And subsequently, these are synthesized in large quantities. One example of that is given to President Trump when they tried monoclonal antibodies specific against the spike protein. But similar monoclonal antibodies are being used against, for example, HIV. They are being used against a number of other diseases where the pathways need to be interrupted. Yeah, may I request the host to start the screen share? Otherwise, it's a little difficult for me to talk about this subject. Is there a problem? Can I assist you in any way?
Uh, Venkat, you have tried to share the screen, that
The
green.
host has disabled the screen, so
Oh,
I'm
I see. The
to,
host should.
Yeah, it started screen sharing, started.
it is
Yeah,
still
give,
coming.
Wait for 1 minute.
Okay, so we'll wait for 1 minute. So sometimes inhibitory pathways can be there in Zoom also.
They can do it. Now they can do it.
right
Try
now,
to do
Venkat.
the screen sharing right now. कौन को screen share करो?
Uh,
Screen sharing हो गया.
okay, let's try. Oops. Yes, indeed, it has
Share
started.
screen.
Yeah.
So I should be able to. Can you see my screen now?
Yes, yes. We can,
Yes,
Venkat.
yes, yes.
After this rather long introduction, I think I need not waste more time on cell injury. This is a standard disclaimer just to let you know that by its very nature, this is a review of literature. It is not meant for personal work. I have tried my level best to give you acknowledge
Screen share नहीं
all
लगता है.
the
कुछ ना
authors,
पड़े ना.
books, and papers that I have.
Connection नहीं की.
I think some of the participants may want to mute their microphone. So I am a cytologist, and I'm happy to say that the father of cytology lived over 400 years ago. That was Robert Hooke, using this extremely primitive microscope. Thankfully, we don't have to use these to look at cells in slices of cork. The father of pathology comes along almost 200 years later, that is Rudolf Virchow from Germany. And he was very famous because he propounded the cell theory. In those days, there was a lot of controversy, and the scientists used to fight a lot about whether cell theory or germ theory is the cause of disease. Now, this gentleman was a very aggressive person. In fact, he even challenged Bismarck to a duel. Instead of pistols or knives, Rudolf Virchow said, "We will both eat sausages. We'll enjoy our meal, but one of the sausages will have Trichinella spiralis. Whoever catches the Trichinella will die, and the survivor is the winner." Of course, Bismarck did not agree with this, but you can understand how much of controversy and debate was there between this. Germ theory ultimately won, and cell theory became relegated to pathologists in our studies because the importance of cholera epidemic, Robert Koch, and tuberculosis took everything towards microbiology. But as pathologists, we have looked at the simplistic cell injury model, that is the hypoxia model, for over 100 years now. And the diagram is so simple. We just have this is the cell, the oxygen is coming. If we cut this, what happens inside the cell? That has been the model of cell injury which we have studied. If you look at a cell culture, you can see here, this is a cell in culture, which when injured loses its microvilli, but instead acquires cytoplasmic blebs because the cell is swollen up. If you look at the normal cell, these endoplasmic reticulum become swollen because of the retention of fluid. And in this normal cell, the rough endoplasmic reticulum as well as the polysomes are aggregated, which become disaggregated in injured cell. This is what we have been studying for the last 50 years. The most important of the cell organelles which can get injured are the mitochondria. So this mitochondria, you can understand that all mitochondria have to come from pre-existing mitochondria only. The nucleus cannot create new mitochondria. And therefore, when the last mitochondrion of a cell is destroyed, the cell has to die. It cannot be reversible. So the pre-existing mitochondria, the telltale sign of the massive damage is the amorphous densities, which can be seen in mitochondria. The other concept which we have very well understood for the last 50 years is free radical injury. Free radicals we know have unpaired electrons which are unstable, and these are the oxidizers. So they oxidize whatever they touch, and they are autocatalytic. That means more free radicals are produced at the end of the injury. The injury is therefore during a propagation reaction. So a typical example of this is peroxidation of lipid membrane. So in lipid peroxidation, there is an initiation where a superoxide cuts the double bond of a PUFA in a cell membrane. So the unsaturated double bond gets cut and the PUFA becomes a free radical with a free electron in it. This is now free to attack other double bonds of the PUFA. So all the double bonds start getting cut and cross-linked like this. So PUFA cross linkage converts what was a lipid bilayer into a onion skin like appearance, which is what we call as the telltale signs of free radical injury. It stops only when you have a termination, which is by beta or other antioxidants to stop the free radicals from propagating. Exactly similar to these myelin figures, cross-linking of proteins also occurs when the sulfhydryl groups of proteins are attacked by the free radical to disulfide bonds. So the proteins are all powerful proteins in nature. Similarly, DVK damage is produced by single and double strand breaks as well as cross linkage. Now, there is a type of new cell injury called ferroptosis, which is a non-enzymic free radical injury. I won't go into details of that because we need to go further. If you look at this, this is what happens in a free radical injury. The CCl4 forms CCl3 and cuts all the double bonds. The double bond is where this leg of this is bent, that gets cut and it gets cross-linked and it forms these myelin figures, which is the sign of free radical injury. Wherever free radicals are generated, whether it is in peroxisome, you have the antioxidant like catalase. If in the cell membrane, that is lipid peroxidation, you have vitamin E. If it is the mitochondrion, then free electron transport. For that, we have vitamin C, vitamin E, and glutathione peroxidase. If it is the lysosome, against that we have vitamin C and beta carotene. So, like that, these free radicals are always quenched. This was the understanding of cell injury when around 50 years ago we found that there is another form of cell death, which is called apoptosis. It was discovered in this tiny nematode worm, which is found in the soil called Cyanorhabditis elegans or C. elegans. It has around 1,000 cells only, of which exactly 131 cells die at a particular precise point by programmed cell death. In contrast, the human body every day 10 to the power 9, that means 1 billion cells undergo apoptosis every day, forming up undergoing the program. We all know how to recognize apoptosis because we are all pathologists. They are these small shrunken bodies with pycnotic nuclei. If you give dexamethasone to a mouse in the thymus, this is a normal thymus, and after dexamethasone, it has all become apoptotic. These are the histiocytes eating this. Here, the electron microscope shows the normal lymphocyte and the condensed nuclei and the cytoplasmic blebs with condensation. Unlike cell injury, the insides of these blebs have condensation, not swelling. Here also you can see a cytoplasmic bleb over here. In this apoptotic, this is a cytoplasmic organelle packed bleb. Here the nuclei have become pycnotic and taken up by the macrophage. Another thing which can happen in cells which have a lot of rough endoplasmic reticulum is formation of these ribosomal aggregates like this. They all aggregate together. In contrast with the ribosomal disaggregation which is seen in cell injury. Here we can see a kidney tubule in which an apoptotic cell is there, and how the nuclear features, the nuclear chromatin becomes clumped at the rim like this. Although we knew about apoptosis for the last 50 odd years, in the last 20 years, a lot of research has gone into identifying and utilizing this apoptosis. And central to this is the identification of the substrates for caspase 3. First of all, the earliest way of detecting apoptosis used to be the tunnel or TDT nick end labeling. TDT labels all nicks which are present in the DNA. Now we can do other kinds of stains, like we can stain for caspase 3. Even a cell which has not started to undergo apoptosis can be found. There's a cardiac myocyte full of caspase 3. So caspase 3 creates other interesting changes. For example, it can activate this phospholipid flipases and phospholipid scramblases, which invert the cell membrane, and phosphatidylserine, annexin, calreticulin, molecules which are normally present on the cytosolic side or in rough endoplasmic reticulum, come outside and we can detect it also. We can either do a confocal microscope to detect it, like shown here, or we can do flow cytometry to detect it. An important thing to understand about apoptosis is that it's an ATP dependent process. There is another way of cell death, which is PARP, which I'll come to in a short while. This PARP also requires ATP. So one of the functions of the caspase 3 is to inactivate the PARP based or the Parthanatos pathway. Other interesting thing include activation of fodrin, which cross-links all the cytoskeletal proteins, activates gelsolin, which causes actin depolymerization. So all the cell microvilli and other cytoplasmic projections are lost. They decrease the cellular adhesion by inactivating the adhesion kinases, and the nuclear and scaffold proteins are also affected by cleavage of lamin, which causes the nuclear shrinkage and fragmentation. Now I have created this extremely busy and complicated slide to explain apoptosis in one slide. So obviously I have to explain it. This square box which is outside, this is the cell. This round over here is the nucleus. This round over here is the mitochondrion. And these are the various things which can get activated in various pathways. So whatever is external to the cell, like TNF alpha or Fas ligand, they cause the extrinsic pathway to get activated, where the initiator caspase 8 is activated. So it could be also activated by cytotoxic T cells, which poke a hole in the membrane with perforin and inject granzyme B into the cytoplasm. This causes direct death by activating the executor caspases, which the downstream of which I've already explained. But the more important part of it is integrated in the nucleus and the mitochondrion. This integration is because of pro and anti-death signals like growth factor or injurious agent. So if the growth factor is not there, then BAX is activated, which causes permeabilization of the mitochondrion. And if the growth factor is there, then BCL2 protects it. Similarly, if there is an injurious agent, it can activate the P53 pathway, which causes a cell cycle arrest. But if the cell arrest does not repair the DNA, BAX is released, which causes permeabilization of the mitochondrion. Subsequent to this, a large complex called the apoptosome gets formed, which is right at the center. This apoptosome is a molecular motor which causes generation of large amounts of initiator caspase 9. PD1 also affects this if this cell happens to be a T cell. So binding of PD1 and PDL1 causes release of these SHP and SHP2, which promote the BAX gene if it wants to kill the cell, or it inhibits the proliferation by negatively influencing the MAP kinase. In the last 20 years, however, programmed cell death has been discovered in a lot of other organisms. The evolutionary biologists have found it in plants and yeast, where we previously thought apoptosis does not happen. Here, the central area is the mitochondrial permeability transition pore. So the mitochondrion is the key. There are no caspases. There is cytochrome C release and calcium influx in mitochondria. There is stresses like reactive oxygen species. And they have divided it into different types, apoptosis like, autophagy like, necrosis like, depending on the speed with which these programmed cell deaths occur. The new concept which is now come up, which is what I wanted to mainly address in the lecture, is oncosis. So oncosis is now thought of as a switching mechanism between necrosis and apoptosis. Necrosis and apoptosis are no longer considered as two opposite ends of a pathway, but that there is in fact an integration which is happening in between. Central to this is understanding of ER stress. ER stress means that whenever there is reactive oxygen species or damage to the cell, there is protein misfolding due to the oxidative damage, and this produces ER stress. If there is mild ER stress, then the survival effectors like chaperones, which are the part of the UPR, which I'll just come to, they get activated and the cell survives. But if there is severe ER stress, then there is hyperactivation and the death effectors get activated. Therefore, the previous concept that the oncosis is a reversible and then an irreversible cell death now is thought of as a way station. From here, it can either go into necrosis or it can go into apoptosis or other types of cell death. The word is derived from oncos, meaning swelling, and is a pre-lethal pathway. The main characteristic of this pre-lethal pathway is the leakage of damage associated molecular patterns and the presence of inflammation, unlike apoptosis. Any sudden shock to cell, infection by rotavirus and other pathogens, cell death, of course, in ischemic injury, they are all examples of this. And in both oncosis and apoptosis can be linked by the energy requirement of apoptosis. A cell undergoing apoptosis can exhaust the ATP supply, and unable to complete apoptosis, it can undergo secondary necrosis. Similarly, if oncosis is inhibited, cellular stress can induce apoptosis in a cell also. So this hypothesis of oncosis, that apoptosis and necrosis are not distinct mechanism but opposite extremes, is the main advance which we have seen over the last few years. And opening of the mitochondrial transition pore is observed in both of these mechanisms. So instead of calling it as apoptosis and necrosis, we now call it as accidental cell death, which is a rapid, uncontrolled cell death. Morphological manifestation is necrosis, Greek is death. It's a major pathway in injuries due to ischemia, toxins, infections, and trauma. It's the inevitable end result of severe damage beyond salvage. It is not regulated by genes. Any specific signals are lacking and biochemical pathways are not there. Happens accidentally when the stress is beyond the limits, and cellular constituents may simply fail or fall apart. Regulated cell death, on the other hand, includes apoptosis, necroptosis, pyroptosis, etc. Many are there. Can occur in the absence of any exogenous environmental disturbance also, but they can also occur in their presence. They utilize a built-in effector, and for development or tissue turnover, these are normal things which are sometimes utilized for cell death. Elimination of useless or potentially dangerous cells, like an infection or tumor, is one of its functions. And the varied inflammatory and histiocytic responses can be there depending on the type of cell death. So in 2018, the committee, there's a nomenclature committee also for this on cell death, met and came up with this long list of, as many hours are there in a clock, so many types of cell death they have described. It's not important to go into each of these. Just important to remember that regulated cell death can have either an apoptotic morphology or a necrotic morphology. So if something is very complex, I like to explain it. But again, the diagram I made to explain the newer concepts in cell injury is a very complicated diagram. So let me explain it. This box is again the cell. This is outside the cell membrane. This is the nucleus, the endoplasmic reticulum, the mitochondria, which are the central actors in this. Various cell signals, which can be death signals or injury signals or ATP loss, all of these converge on a cell to cause changes in the cell membrane and a signal transduction or a membrane dysfunction. These can activate by an integration process. The two main integrators are the mitochondria and the endoplasmic reticulum. The mitochondrial membrane can be stabilized or destabilized. And if it becomes destabilized beyond a point, then it releases cytochrome C, which causes formation of this molecular motor called apoptosome, which shuts off apoptosis. This is an ATP dependent process. Another ATP dependent process is because of nuclear injury. It doesn't involve the cytoplasm at all. It is a process called Parthanatos, where when there is nuclear damage, an ATP dependent cell death occurs. What ER stress and other kinds of injurious agent can do, like pathogen associated molecular pathways, is either the necrosome or inflammasome activation. If the necrosome is activated, it causes a necroptosis. If the inflammasome is activated, it causes a pyroptosis. These are molecular motors very similar to the apoptosome. In contrast to apoptosis and parthanatos, where nothing happens outside the cell after it is triggered, necroptosis and pyroptosis release both DAMs, that is damage associated molecular patterns, as well as interleukins and cell signaling molecules, which cause inflammation and necrosis beyond that cell. So apoptosis is within the cell. Necroptosis and pyroptosis go beyond the cell to injure the tissue and the entire organism. These complex molecular motors are illustrated here: the apoptosome, inflammasome, the necrosome, and another one called the proteasome, whose function is to degrade all the proteins. So the important thing here is the ER stress. What is ER stress? ER stress means oxidative damage to the proteins. The proteins get injured. Whenever a protein gets injured, it has a tendency to misfold. This misfolding causes problems, and therefore proteins called chaperones, which include the heat shock proteins, come and bind to these proteins to protect them. Stress proteins are rescued by this mechanism, and the function of the protein is retrieved after the misfolding is refolded. But if the protein is irreversibly damaged, another protein called ubiquitin binds to it and marks it for later proteasomal degradation. So here you can see, this is the protein, heat shock protein. If a protein is misfolded, then it binds to the chaperone. Some chaperones are called holdases. They just hold it in position for other things to come and act. These others are the ones which cause a repair of these misfolded proteins. So when it binds to this is a misfolded proteins. Other proteins like GROES7 will come and they will bind to this area and refold it. So essentially this is an ATP requiring process, energy dependent, unfolding and refolding. So the protein is unfolded, ejected through one area and refolded. The protein has a tendency to automatically reassemble into its original folding if the misfolding is unfolded. So the holdases and the unfoldases are the essential components of this chaperone mediated retrieval of misfolded proteins. If these proteins cannot be so unfolded, then ubiquitin binds to it and degrades it in the proteasome complex. Misfolded proteins are a pathognomonic feature of so many things in pathology, whether it's Russell bodies, Mallory-Dunk bodies, Kuru plaques, neurofibrillary tangles, Lewy bodies, Huntington's inclusion. All of these are representing misfolded proteins. Now, ER stress is very directly linked to apoptosis. One good illustration for this is to understand how calreticulin functions. Calreticulin is one of the chaperones. It is not just a chaperone, it is also a calcium binding protein, and it is present in the ER. Whenever the oxygen mediated damage is there in the endoplasmic reticulum, there is increased expression of calreticulin, which is expressed in the endoplasmic reticulum. But if the damage becomes too much, then there is a relocalization of the calreticulin. It goes from the endoplasmic reticulum to the cell membrane. And once it goes to the cell membrane, it now acts as a phagocyte marker for taking up of the apoptosis. It also triggers apoptosis because of the ER stress and activation of the other pathways of apoptosis. Let's look at another way of cell death, which is pyroptosis. So pyros means fire. You call it pyroptosis because the cells look like they are flaming out. One cell is just flamed out. Inflammation has happened in just one cell. The other reason why it is called pyro is because pyrexia, they produce fever. What happens in pyroptosis? This can be thought of as programmed cell death with collateral damage and inflammation. It is seen in macrophages and epithelia infected with Salmonella and Shigella. These are caspase dependent, but not the same caspase as apoptosis. Instead, this is a caspase 1. So caspase dependent, but pro-inflammatory cytokines are also released. What happens is that nod-like receptors, that is NLRC4 or toll-like receptors, recognize PAMs present in microbes. These activate the nod-like receptors to assemble the inflammasome. The inflammasome is made up of this NLRP3, which has this card or the caspase activation domain, whose function is to give pro-caspase to caspase 1. Caspase 1 does two things. It releases interleukin 1, which causes inflammation and fever, and it releases this Gasdermin D. This goes to the cell membrane and pokes holes in it, forming pores. Therefore, if there are pores in the cell membrane, the cell is going to swell up and die. So there is flow of ions, swelling and lysis of the cells, and release of further dams. So the damage associated molecular patterns cause further inflammation because of this. But the nuclear integrity is maintained, unlike apoptosis. There is no fragmentation. The caspases are different. The second type is another necrotic morphology type of regulated cell death, which is necroptosis. Here, the triggers for the necroptosis are almost same as for apoptosis, like reactive oxygen species, DNA damage, metabolic imbalances, or because of activation of dead domains, pattern recognition receptors, toll-like receptors, DNA binding protein, zDNA, ZBP1, all of these which cause apoptosis can cause necrosis when caspase 8 is inhibited. So an apoptotic signal, if it finds that the caspase 8 is inhibited, can cause necroptosis. What happens in necroptosis is there is sequential activation of proteins which assemble to form this necrosome. So there is this RIPK3 to which this MLKL will come and bind, and this forms a ripoptosome. This MLKL, like Gasdermin, can form pores in the cell membrane and kill the cell. So this ripoptosome is like a rheostat between apoptosis versus necroptosis, depending on whether caspase 8 is available or blocked. Some infections like CMV can actually have caspase 8 inhibitor to escape from the cells lysing and the virus getting lysed along with it. Here they have illustration of the same. This is apoptosis when caspase 8 is activated. If this is not possible, this is the alternative pathway where the MLKL assembles to form this necroptosis and the ripoptosome. Now we come to yet another form of cell injury and cell death, which is called Parthanatos. So Thanatos is the Greek god of death. So hypnos, if you touch somebody, the Greek god of hypnos, if he touches somebody, he will fall asleep. And if the Greek god Thanos touches somebody, that person will die. And that is the derivation for the Marvel super universe villain called Thanos. You can remember it like that. So it is Parthanatos. And the reason why it is called so is because this is a cell death pathway activated by this PARP1. PARP1 is poly-ADP ribose polymerase, a nuclear enzyme, which senses mild DNA damage and causes repair machinery to get activated. But if there is profound DNA damage, it causes a PARP1 mediated cell death. This is because the PAR polymer accumulates and causes translocation of AIF protein from mitochondria to the nucleus. This causes large-scale DNA fragmentation and cell death. So nuclear condensation and fragmentation is present, but no cytoplasmic change is produced. This is an ATP-dependent process and is extremely important in stroke, neurodegenerative disease, spinal cord injury, and a lot of research is going into this. This is just an illustration of Parthanatos, where active PARP releases the PAR polymer, which causes relocation of the MIF and AIF back to the nucleus to produce DNA fragmentation. Let's come to some examples and study some diseases where these kind of ER stress are now being evaluated. Diabetes is a very important example. In diabetes, the beta cells are lost in type 1 diabetes. In type 2 diabetes, you have resistance to insulin. And now we are discovering that ER stress is at the base of both of these processes. What happens? When there is ER stress, there is nuclear gene upregulation, which was discovered way back 30 years ago by the UPR in yeasts. And we have just discussed how chaperones are mediated for the protein unfolding to be cured. Apoptosis is triggered if these fail. When there is ER to nucleus signal transduction abnormality, you start getting diabetes. So if there is abnormal expression of ATF, PERK, IRE, these are various signal transduction methods between the endoplasmic reticulum and the nucleus. So mutations in these in rodents can produce experimental diabetes. Similar mutations in rare human analogs have also been described, like abnormal nuclear translocation of CHOP. Sometimes you get misfolded insulin, sometimes you get it in obesity. All of this, the end result is that apoptosis of beta cells is triggered. So there is an injury which is because of this oxidative stress and ER stress. The autoimmune process which we stress for generation of type 1 diabetes is mediated through the oxidative and ER stress. And the importance of this is if you can block the pathway, you can prevent beta cell damage and destruction and preserve some of the beta cells for continuing to produce insulin. And a lot of drugs are being tried in this respect. On the other hand, we have lipotoxicity and glucotoxicity. That means when the blood levels of lipids and glucose go up, it causes a stress response in all the tissues of the body, all over, everywhere. So glucotoxicity and lipotoxicity are responsible for so many of the changes which we see in from the eye to the kidney to the heart. It is all related to stress related cell injury, ER stress. And so now that we are working out some of these, we are trying inhibitors to each of the pathways to see which one will have an effect. For example, you have now ER stress modulators. One example, an experimental molecule called azoramide. What it does is that it reduces beta,
IAPM में बोल
and it can also protect tissues from hyperglycemia, hyperlipidemia, and it can prevent this diabetic cardiomyopathy also. So that means that these drugs not only keep the beta cells active, they reduce the damage which is present in diabetes also. So in the short term, the UPR activation aids in restoring the protein production and folding balance. But if there is chronic ER stress, then there is apoptosis and autophagy getting triggered. We have not discussed autophagy. I will cover it in just one slide. Important is that the ER stress is what causes the cell damage in a pleiotropic fashion in diabetes. So what is autophagy? Autophagy is a tightly regulated set of genes called the ATG, which include genes like Becklin, LAMP, and RAP7. These are upregulated during any cellular stress, like chronic ER stress. Whenever there is deprivation of nutrients, where deprivation of growth factor, these ATG genes the balance is changed. The end result is recycling of organelles, extensive cytoplasmic vacuolation, phagocyte uptake, lysosomal degradation of the organelles, so that the constituents are released for the cell to reutilize for its own purposes. Is there an autophagic cell death? Probably not, because this is a mechanism which is always activated when there is cell injury. And if injury beyond a limit causes death, your ATG genes will always be active. But now we have slowly discovered in plants, definitely there is autophagy mediated cell death. And slowly we are realizing that neurodegenerative disorders also, autophagy mediated cell death is important, especially stroke, which is a very common disease. Similarly, there is great potential in chemotherapy. Inducing autophagic processes can help us target neoplasia. Let's come to another example, that is gout. We have all seen this is an aspirate from a gout, and it's got these crystals. There is a giant cell reaction to the crystal, which you can see. Occasional histiocytes you can see. Some neutrophils you can see. If we polarize it with cross polars, you can see so beautifully needle-like crystals are birefringent because there is deposition of monosodium urate. We always thought that it was a simple disease, that when there is gout, there is hyperuricemia and the uric crystals get deposited and cause inflammation. But it turns out that there is very complex pathway involved in pathogenesis of this disease. So if anybody has gout, what happens is if he drinks too much or eats a lot of meat, you get a reaction. That is, there's a flare, amplification, and then resolution. So for a week or so, his toe becomes swollen and is extremely painful, and after some time it goes away on its own or with anti-inflammatory drugs. What happens here is that this flare initiation starts because monosodium urate is actually a DAMP, or a damage associated molecular pattern, which is recognized by toll-like receptors as well as by endophagocytosis or lysosome formation, which activates the inflammasome activation. Once inflammasome is activated, there is a lot of interleukin 1 release, which potentiates the inflammation. So once the flare starts, then there is a problem. Then there is amplification. All the NF-kappa B activation causes release of all these mediators, including interleukin 6, TNF alpha, interleukin 1 beta, all these get released. And it so turns out that if you use a monoclonal antibody against this interleukin 1, it can cut the flare initiation at its induction itself. So by understanding this molecular mechanism, we find a way to cure it. How does it terminate on its own? Subsequently, the anti-inflammatory molecules like TGF-beta starts getting released. Then cytokine-inducible SH2 suppressor, another inflammatory mediator which inhibits inflammation. Then suppressor of cytokine, melanocortins, ACTH. Release of these mediators causes the inflammation to go down. Interestingly, the reason why we get a gouty tophus is when there is a chronic foreign body granuloma formation, which is initiated by another form of cell death called netosis, or neutrophil extracellular trap-induced cell death. When netosis is activated by the monosodium urate crystals, this net forms a network of the nuclear contents of the neutrophil, which traps the cytokines and slowly degrades them. And so when you look at this, you can find that there are a lot of histiocytes which are there in between the crystals. We can find a lot of inflammatory cells which are here, neutrophils. And these are all because of the inflammasome associated disease pathways. The last thing I would like to talk about is viroporins. Not a lot of us might have heard about this because the microbiologists are the ones who mostly talk about these. What are viroporins? These are viral proteins which modify cell membranes. Their aim is to permeabilize them so that the cell entry is gained by the virus, or release of the virus is mediated, or the endoplasmic reticulum is modified for creating lots of virions by RNA viruses. A good example of it in a DNA virus is HPV E5 oncoprotein, but I'd like to talk about SARS-CoV-2, which has two viroporins called E protein and 3A protein. So this is the cell. Here we have the cell, here we have the endoplasmic reticulum, and here we have the nucleus. What has happened is that the virus has come, attaches to the converting enzyme 2, gets internalized, and then it releases the viroporin, which is the 3A, for example. If it is 3A, it goes and permeabilizes the endoplasmic reticulum, which is its main function. But it has a second function. It activates the NF-kappa B. NF-kappa B goes into the nucleus and releases a lot of interleukin 1 in the form of pro-interleukin 1. And this 3A viroporin also activates the procaspase 1 to caspase 1, that is the inflammasome pathway, to create lots of interleukin 1, which creates the cell injury, because as you know, caspase can activate gasdermin and poke holes in the cell membrane, and it causes inflammation. So the inflammatory storm which is created in SARS-CoV is because of this inflammasome activation. So necroptosis, if you look at it, is also activated. Therefore, when the SARS-CoV-2 comes, it can either cause apoptosis of the cell, or if the apoptosis is inhibited in any way, then it starts releasing a lot of this P17 and the MLKL, which causes necroptosis, which also cause the death of the cell which is infected as well as inflammation. So the activation of NLRP3 by the NF-kappa B pathway causes the inflammasome activation procaspase 1, which potentiates the cell death and the inflammation which acts. What is the importance of this? We have used drugs, all these drugs which we have tried out, many of them you might recognize, including something like even hydroxychloroquine. Hydroxychloroquine inhibits the lysosomal fusion. IL-6 inhibitor like tocilizumab is because it blocks the interleukin 6 release, which is over here. So the interleukin 6 is blocked by this monoclonal antibody. Almost all of these drugs have been tried either in cell cultures or in animal models or in even human trials to see whether we are able to cut the inflammation. So by understanding the inflammatory processes which have recently been described, we are able to target new diseases also and old diseases also. So to summarize, the cell injury pathways are now known to link the cell membrane, the endoplasmic reticulum, the mitochondria, and the nuclei. And each of the topics which I have talked about today is actually a topic for a full-fledged lecture because there is so much detail which is known. There is signal transduction in both these directions. And all of these pathways are targets for drug development. The very active research, as I said, there are innumerable number of monoclonal antibodies and inhibitor drugs which are being tried all the time in cell cultures. They are important for inflammatory diseases like viral infections, typhoid. I've not touched on typhoid also, gout, arthritis. These are all very good examples of how we target the new cell injury pathways which we have described. They are also very important cell loss mechanism. They are cell deaths. So beta cell loss in diabetes, cell neurons loss in neurodegenerative diseases, all of these are mediated by the kind of newer cell injury mechanisms and cell death mechanisms which have been discovered in the last 10 years or so. These are the references which I have used. The other references were referred to in the slides themselves. And I would like to end this talk with an acknowledgement that I borrowed a couple of slides from my colleague, Dr. Aruna Nambirajan, and a couple of slides which I showed were from presentations by our residents, Hina and Christabella. Thank you all very much for your patient listening. I wish you all a good evening.
Thank you, Venkat, for this wonderful talk. And it clearly shows how things have developed over the past 2 or 3 decades. From what we know of necrosis and cell injury as undergraduates, things have gone become more and more complex. We also know that ATP is required even for cell death. And earlier we used to think that ATP has to go and only then the cells will die. But and there are so many different varieties. The importance of these is that nowadays the development of drugs and molecules have also gone hand in hand because we have understood this complex pathogenetic mechanisms. And that is why even a simple thing like gouty tophus, there are different molecules that are being tried to stop the um, inflammatory process and the cell death process in the middle. So I'll not go further. Venkat, I'll open it for questions from the audience. The best part will be that if you can just type your questions, maybe Dr. Asaranthi and others from there can just read out those questions, or I can read out and Dr. Venkat can answer those questions. So if there are any, please feel free. It's a complex topic, I know, and many would not imagine that such things have developed over the past several years. Any questions from anybody? Anybody would like to? You can raise your hands, and Dr. Asaranti at the headquarters will allow them to talk or anything.
We can invite questions.
Yeah, I've invited questions. Anybody can either they can write in the chat and we can read it out, or if they want to raise their hand and
I think not. I don't think there's any question coming up. Dr. Gupta,
okay.
I like to congratulate Dr. Venkat. It's so much of complex topic, and he has condensed them and make them very simplified to be able to understand to anybody who is listening to it intensely. And so I think many of the residents possibly had understood this quite well, and they are more familiar with all these various genes and various enzymes. And what I all the time feel is because this apoptosis, it was described only in 1972. But you know what I feel is if we look back into the embryology of development of ventricles, Jesus say that there is cavitation which takes place. And when it was described, it was more than 100 years back. During that time, the word apoptosis was not there. So apoptosis had been coined much later. And so it possibly, if apoptosis was already existing, this term during that time when cavitation of the heart or the ventricle was described, possibly the word apoptosis would have been used. That's what I feel very strongly about. And thank you very much, Venkat, to make it so simplified for such a complex topic, and which is possibly the right kind of topic which has been chosen. Thank you very much, Dr. Datta Gupta, for choosing such kind of good topics.
Thank you, Dr. Kim.
Yes,
Thank you very
excellent
much.
lecture by Dr. Venkat Iyer. Really, it was very nice, and he had simplified the very complex topic. And all these topics are very important, though they appear to be very basic topics. But as we go for our day-to-day diseases which we encounter, and hand-to-hand, very molecular targeting and treatments are coming up. As he showed that for diabetes, for gout, all the if you know these details, then we are getting all those treatments are developed. And the main thing was that it was a very complex topic, but he has expressed it very well with all those clear and lucid diagrams. So once again, thank you, Dr. Venkataya, for such a nice and excellent lecture and presented so well. Thank you, Dr. Venkat.
Dr. Vatsala, there's a
Yes, I also
there's one Dr. Vijayalakshmi has asked one question, and
Yes.
that is, Venkat, if you could just clarify on the beta cells in diabetes. She has asked this question. Sir, please tell us about beta cell death, and on this, if you want to.
So that's actually one of the most complex areas of research which is going on currently. We know that there is a autoimmune reaction against these beta cells, which is what initiates the process. But once the initiation of the process takes place, subsequently there is amplification of the cell death. And this amplification, we cannot really stop the initiation of the cell death. Our aim, what is our aim? We want to treat diabetes by preventing beta cells. Even if you transplant beta cells, if a person is in a IDDM, the beta cells can be destroyed once again. Our aim is to cut the intermediate steps which are involved in the initiation of cell death by an autoimmune process, which is initiated by numerous viral infections and other causes. There are inherited causes also, but they are rare. More commonly, you have a viral initiation. or a idiopathic kind of initiation autoimmune process is starting. But once it starts, we want to stop it from further damage. And therefore, we are trying to elucidate the steps of this damage, look at the cell injury process in detail, work out the pathogenetic mechanism step by step, and see if any particular pathway is more important. And if we inhibit that, we can preserve the beta cells even if the initiating event cannot be necessarily stopped. We may not be able to stop the initiating event, but we can cut, just like rheumatoid arthritis, the initiation is autoimmune, but the response is to delivery of a monoclonal antibody against the intervening molecule which amplifies the inflammatory response. Similar thing we tried in the beta cell destruction in diabetes.
Thank you, Venkat. There are none. I hand over to Dr. Vatsala and Dr. Asarantikar. Thank you very much. Thank you once again.
Yeah.
Thank you.
Thank you so much, Dr. Venkat. You really justified your duty to the topic like newer concepts and many things you have told, which I think the PGs must have not had earlier, and it will be really beneficial. I can give you the feedback of this topic after I discuss with the PG postgraduates, and you have prepared it in such a small time with the request of Professor Datta Gupta. So we are really grateful to you. And it was a real very nice, and I have got so many feedbacks from others also. It was very good, and you explained it so well. Thank you so much.
Thank you very much, ma'am. It was indeed a great honor to speak for the IAPM.
Okay, Asanti, this was our start
Thank
of
you.
our webinar series, and I think it is excellent. And we hope to continue further with such topics, basic topics covering. I once again thank Dr. Venkataya for such a nice presentation. And I hope that all the audience which were there, who were logged in, they must have enjoyed this lecture and learned many basic things regarding these basic processes
madam.
which are important.
One
And,
question has
once-
come, I think. Madam, please wait. Please
Yes.
elaborate on ferroptosis. One question has come. Dr. Iyer,
Yeah.
please elaborate on ferroptosis if possible.
So ferroptosis is cell death when iron levels inside the cell. Am I audible? Yeah. When iron levels inside the cell are very high. What happens in that is that there is a now any transition metal has the ability to exist in more than one ionic state, and therefore it acts as a generator of free radicals, of free electrons, unpaired electrons. So Fe2 plus 3 plus, this transition means that an iron molecule can generate a free electron which can cause free radical damage. What happens in ferroptosis is that a protein iron complex gets formed, which becomes a free radical generator by a non-enzymatic catalysis. That means that the people have modeled the protein iron complex which gets formed and found that a iron catalytic site gets created, which acts like an enzyme. So it is one of the few examples where in a biological system, a non-enzyme is catalyzing a reaction. This catalysis creates free electrons, which creates free radicals. Once these free radicals get generated, cells which have, typically this is seen in hemochromatosis, hemosiderosis, iron overload, hemophagocytic syndrome. Wherever iron is overloaded, whether it's in the macrophage, whether it is in a parenchymal cell, it kills the cell by ferroptotic process. The iron, you may try to chelate, but because it is an autocatalytic process, so the amplification by even small amounts of iron, which are still not removed by the dysferrioxamine or whatever chelating agents which you give, they can continue to cause cell destruction. So to inhibit this, you use a drug which specifically binds to this iron catalytic site. And if you can inhibit this autocatalytic site by using a drug which binds specifically to it, you can prevent something like liver cell damage from occurring. And that can, as you know, that you can get cirrhosis also, you can get liver damage. And these are all, people are trying these kind of experiments in vitro and in cell cultures to see whether we are able to utilize our understanding of ferroptosis and cell death to prevent the liver cell injury or the macrophage injury, which underlie so many of our iron overload syndromes, thalassemias, etc.
Thank you, Venkat. Yes, Dr. Kar, I think. If there are none, please unmute Dr. Kar. Dr. Asarante, you can unmute.
Yes.
Yes, sir. So how we have planned our webinar is one general pathology topic alternating with one slide seminar, like we had discussed earlier. So our next webinar will be next Friday, 7 p.m. and it will be on tumors with epithelial morphology. The speaker will be Professor Bharat Rekhi. So I will again send the link and everything.
Thank you. Good
Okay.
night
Yes,
to everybody.
Professor Vikyananda is there. He wants just to give blessings to you all. Professor Vikyananda,
Okay.
just
Please.
be in line.
Thank you, sir.
Yes.
Namaskar,
Thank
sir.
you.
Thank you, sir.
Namaskar, sir. Thank you, sir.
Thank you.
Oh,
Thank you very much.
Thank you.
Thank you, sir.
Thank you.
thank you. So I think we will move on to our next webinar on next Friday. Hope that will again.
Absolutely ignorant of what has gone within this long period of this. So thank you very much. Blessings to all of you young people, particularly Siddartha, the pioneer in the scientific sessions
Sir,
which you are
no,
having.
sir. We have all with the
Yeah.
Thank
And
you, sir.
your president, girl president, only I wish he loses little bit of his weight. That is all what I expect.
Who, sir?
Thank you very much.
Sir, Professor G.N. Mahante. Sir, thank you, welcome. Thank you, welcome.
Actually, what happened when last meeting was there, I just suggested Dr. Asaranthi that why don't you take the chance to make the whole year active and you arrange some programs which will benefit the postgraduate students. And I am very happy. The first lecture started with the first chapter of the general pathology, a cell injury. So I was teaching general pathology and cell injury. I'm very happy. And I just thank Dr. Iyer, Dr. Gupta, and Madam Baschala, Dr. Asaranthi for doing all the best. I wish this thing will continue in the future. Thank you.
Thank you all.
Thank you, sir.
So thank you all. Should we close now?
Yes.
Anyone has to tell anything?
Yes, yes.
By this, sir, Dr. Pradeep is there. You want to tell anything, Dr. Pradeep?
No, nothing.
Because you were one part of the, while selecting the topics, I think Pradeep have told me to go in this way so that it will be unique for the postgraduates.
Thank you.
Thank you.
Thank you. Dr. Das.
Okay. I think.
So, madam, should we stop now? Should we stop now?
We can close now. Okay, and we'll meet again next Friday. Everyone. Okay.
Yes, madam.
Yeah.
Thank you. Good night.
Thank you,
Good night.
everyone. Good night. We will meet again on next Friday.
Thank you, Ashim.
Yeah.